Hello and welcome to another episode of Biohack Chat. I am your host, as always, Justin. Welcome to the show. Today's gonna be a fun one. It's always a fun one. What am I saying? They're always fun. I have fun. That's what counts. So today we're gonna be once again chatting with my good friend Gabriel Licina. This time we're gonna be talking about something that we actually talked about on an earlier stream. So this is going to be an update to show the progress that he's made, talk about changes and this kind of thing, and how it's going. I think it'll be fun. It'll also be, if you didn't see the last one, we'll go over all the specifics of the project and all that kind of stuff. So quickly before we get into it, you'll notice that the donation bar from last time is full. This is super exciting. So as you can probably tell, we've met our goal, which means that the Plasmid that we designed over the last two streams is actually going to be able to get made. So in about a week, I'm leaving it up on GitHub. There's links in the description. In about a week, I'll actually be putting in the synthesis order. So I'm basically giving it one more week so that if anybody can find any issues with it before I submit it, kind of now is the time. So be sure to do that. Other than that, let's get right into it. So, without further ado, allow me to introduce my good friend Gabriel. And you should be live. Oh, no wait. It's the other one. There you are. Now you're live. Okay. Say hello.
Hello. Hi. So, here I am again. My name's Gabriel Licina. I work for a 501c3 nonprofit called Sci House. And we do genetic engineering and environmental remediation, I guess. And we've been working on a really cool project. And we mentioned this project a couple of live streams back, right?
Yeah, I think this was the first or second one that we did as a Biohack Chat. I'd have to check, but yeah, it was a little while ago. It's been a hot minute.
Yeah. So. This has to do with genetically modifying bees. So once again, I will just throw this. Here's the folder for those of you who want to play along.
Yeah. So just in the live chat,
there's not only the papers, but also this has all the bee stuff. So it's got the genes that we've developed. It's got the supplemental materials. It's everything you need to know about Bee Project 2020.
Yep. Also, the Plasmid itself is also on GitHub. So yeah. There's multiple different ways for you to get this DNA if you want to attempt to replicate this project, for those who are interested. And yeah, because I mean, both me and Gabe are very big on the open source thing. So everything we do is open source, unless we have some sort of horrifying contract that says otherwise. But we try not to do those. Anyway, so do you want to give them the short version of the Bee project? And
yes,
then we'll get into the specifics.
And then we'll get into the details. So the short version here is that somebody did some research, and they found out that if you genetically modify the gut bacteria, specific gut bacteria, they're called S. alvi. I'm not even going to attempt to pronounce the first name. I would just butcher it. So it's S. alvi.
Yeah, it's definitely not Snodgrassella.
I think it is.
It is.
I don't know if those two L's are pronounced as an L or a Y.
Point is, it's some dude's name or dudette. I don't know. It's someone's name, and
it got turned into a bacteria name. So anyway, it turns out that the genes that are in this bacteria can also be expressed by the host organism. So this is like a whole nother level of crazy. How do you genetically modify an organism? Genetically modifying fully formed organisms is actually very difficult. Because you have to usually get into every single cell. This is why we use AAV when we're dealing with mammals, because you have to basically make a virus that infects the whole animal. But for whatever reason, bees are able to do this cross-pollination. It's going to be bee puns all the time. Sorry.
I think the technical term would be horizontal gene transfer.
It is a form of horizontal gene transfer, where they can take genes that are in their gut bacteria and then express them in the bee. And so this is really cool because it means that you can isolate some of these bacteria, genetically modify them, and then reintroduce them, and then through this mechanism, modify the bee.
Yeah, and modifying bees without this is actually really hard. So
yeah,
we have one friend who attempted this, and his solution was you basically get bee eggs, which if you've ever seen a bee egg, they're not the largest things ever. They're pretty tiny and delicate. And I mean, so typically when people do insect modification, they start with eggs and then they do what's called micro injection. So they literally make a teeny tiny needle and they just like poke the genes into the thing. But like, there's more than a 50% chance that this A pops the egg because you just poked a hole in it, or B, yeah, no.
So this modification.
Yeah, or yeah, okay.
So this modification allows the bees to be resistant to the problems caused by neonicotinoids, which are a primary element in a lot of pesticides, and which has been directly linked to colony collapse disorder.
Yeah. Which, for those of us who are not familiar with this, colony collapse disorder is awful. It's basically, the short version is, for seemingly no reason, you'll have a perfectly healthy colony of bees just basically all die. And so this has been happening on really horrific scales. So there have been like literally thousands and thousands of hives that are just like dropping dead. And for those unaware, bees are really important. We need those.
Yeah. And on top of this, we also have all of the bees that we don't normally think about. So like when we think about bees, we normally think about bees and hives and honeybees, which is like a European bee. Yeah. But actually, like most of the bees and most of our pollinators are called solitary bees. And so we're still having these problems, but it's not like a colony collapse in as much as they're solitary bees. But the issue is still there, which is that you just have bees that are getting lost and dying. And so this is to avoid that, to make them immune to the chemicals that are causing them to die. And so step one was, of course, to find, like, sure, I could go and isolate the bacteria myself, go out, like, hunt myself a bee with a very tiny spear. But
just a little,
that sounds like crossbow. That sounds like a lot of hard work, and I don't like hard work. I'm lazy, and so I went to go buy this bacteria, and it turns out that the company that sells, basically predominantly it's a monopoly. Ugh, words, they're a thing. They're not selling it. You need to prove that you're working on some sort of project that is protected by a patent, I think. And so then we were like, okay, back to doing it the hard way. And so we went out and we found ourselves a bee. And then it's really interesting because it's like bee bacteria and it's gut bacteria, so it's internal, and it happens to luckily, thank God, have a innate immunity to a certain type of antibiotic called oxytetracycline. This means that if we have, so the media we have here, dun, 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 is a combination of LB agar, which is your normal bacteria media, but with 5% fetal bovine serum, because living organisms. So it's a mashup between bacterial agar and the stuff that I use for doing mammalian work. It's actually kind of weird.
That is super weird.
It is.
That's like the Vibrio fischeri. You're supposed to add beef extract to the media.
Exactly.
That's real weird.
Yeah. And then on top of that, we also have our oxytetracycline. And then we also incubated it in a CO2 incubator. And so the combination of all of these different factors means that we're basically killing off anything that isn't the thing that we want. And so we actually managed to get success. And so these guys are going to be sent off to a secondary laboratory because having a second pair of eyes on your work is always really good. And we're going to get them sequenced. Sequenced? We're going to get them sequenced. To make sure that they're exactly the thing that we're looking for. And then we're going to modify them. So, cool. But this is really great. You know, we've managed to isolate that, and it wasn't even that hard.
Well, I mean, you had all of the right materials.
I had all the right. Well, yeah, but I'm just saying.
It's like, it's not hard to get to space. You just need a rocket.
Exactly.
It's like, well, I mean, technically. But that's pretty cool. So I mean, the fact that you isolated it is awesome, especially because there's no restrictions on what you can do with that bacteria now. Like, there's no, like, fuckhead with a monopoly being like, you're not allowed to share these. You must sign my material transfer agreement. You're like, I got this from a bee. Would you like my bee transfer agreement? The agreement is, here, take my weird bee bacteria.
Yes.
So that's
it's the BTA, the bee transfer agreement.
BTA. So that's really cool. I mean, this was actually the part I was probably the most skeptical about. Like,
yeah, no, I remember you were like,
I was like,
this is never gonna, you're gonna spend 6 months doing this.
Yeah, and I'm both impressed and surprised that it just worked. So that's pretty freaking dope.
Thank you.
So now that so now you've got the bacteria, you're gonna get it sequenced, and here I'm gonna pull up a picture of what this bacteria looks like. Also, since we're talking about bees, might I introduce you to Dr. Bees? Yeah, if you haven't seen this short, actually it's a pair of shorts by Harry Partridge. I would highly recommend it because it's fucking hilarious, and Harry Partridge is dope. Okay, so let's have a look at Smogracella alvi. So, I mean, it's a pretty normal looking bacteria as these things go. Oh god. What the hell? Okay, Chrome decided that it was not having it. Okay, so, I mean, it's a fairly normal looking bacteria. It's rod shaped, as you can see. Yeah, I mean, the interesting thing is that it is able to do this weird horizontal gene transfer thing. It also has a very interesting metabolism. It can eat all sorts of different things. But I mean, beyond that, it's fairly typical. It's mostly found in bees. There's a tiny.
It's one of their primary gut bacteria, actually.
Yeah. There's an interesting little picture here of somebody who's dissected a bee, which just sounds extremely tedious and difficult. But so did you actually have to like dissect out the gut, or did you just like blend it and put it on a dish?
So I went for what I feel like is the most responsible way to do this, which is lots and lots of negative controls. So first we dissected the bee. Then we broke the gut open. Then we yeeted the entire thing into a bottle of LB. And so we just let everything go bonkers.
Okay.
Then we had one plate that had no antibiotics but was going in the incubator. We had another plate that had antibiotics and was going in the incubator. Then we had a plate that had no antibiotics and it was outside of the incubator. And then we had a plate that had outside of the incubator and had antibiotics, so that we could actually like track. So if anything didn't work, you could look at one of the plates and say, oh, like this is where it's fucking up.
Okay.
Because, that's how you do experimental design.
Okay. I mean, that makes sense.
Yeah. And so
yeah, you're right. It definitely does seem to be looking at this graphic, like there definitely seems to be a very high proportion of the S. alvi.
Yeah.
That's really interesting.
Yeah. Tremendous amount. I think it's actually the reason that they used it because it's so prevalent. I don't
Interesting.
necessarily think that the transferring is. Yeah, so here are other plates just to see like the bonkers amount of growth. If you're not doing all of the restrictions, it just goes crazy. Can
Okay.
you see this?
Yeah, I can see that. I mean, there's a lot of growth on all those plates.
Right, because so each step of restriction caused a different decrease or change in the amount of growth.
Weird.
Yep.
Very weird. Okay.
Just
So,
a quick aside, somebody
yep.
was talking about OpenMTA. OpenMTA is awesome, but fun fact, kids, there's a clause in the standard MTA that is anything that is part of a open gene library. So like everything that I ever receive, I add to a library and then I make accessible to everyone if they ask for it. If something that is protected by the normal standard MTA is in an open library, being in the open library both violates but also cancels a standard MTA, meaning that by breaking the rules, you make the rules non-obligatory.
Yeah.
It's
I think
a weird
it's
loophole.
one of those, like, once it's out, they can't do anything about it.
Yep,
Like,
it's a weird loophole, so exploit that shit.
basically. Yeah, that's pretty good. Okay, so you think that it's not that they chose this bacteria because of its ability to do the horizontal gene transfer, just it happened to be the most prevalent thing?
Well, this is what's making me really excited about this project, because this is just step one. Okay, if it's bacteria specific, is it specific to this bacteria? Because this is all just kind of fairly new research. If that's the case, then we need to look more into how that mechanism works. But what if it's bee specific? In which case, we need to look more into how that mechanism works. But that means it's coming from a different position.
Right.
Currently, the theory is that it's mostly bee specific, but nobody's actually looked at this in other organisms, because this is like a whole new thing that nobody's really looked at.
Why do I
So
feel like this
what
is going to end
if-
with you, like, snorting bacteria and then chugging some neonicotinoids? Ah, God damn it, Gabe. Why is it always this?
No, I'm just saying.
Don't snort the
saying.
bacteria, you psycho.
I'm just saying, what if this mechanism is actually preserved across multiple species?
This is going to turn into neonicotinoid rats.
No, this would be one of the most fundamentally cool things that could be done in genetic engineering. What if you didn't need an AAV?
Yes, no, I agree.
To modify an organism. What if you could modify a fully grown mammal by genetically modifying its gut bacteria?
That could be very cool. I mean,
that would be very cool,
well,
but nobody's explored it.
Yeah, and I mean, there's some very interesting research where, or it's not even research, it's just case studies where people will get a bone marrow transplant, and then over time, for whatever reason, cancer, leukemia, whatever, but then over time, none of the cells in their that are like easily sampled in their body contain their DNA. Like eventually they contain all of the DNA of the donor. So I wonder if there's a similar connection in the gut. Like if you can modify enough of the cells, because I mean the gut is extremely vascularized. So if it's constantly like bleeding these genes into the rest of your system, I wonder how far it would spread.
Right. And then this also has to do with, so of course, I mean, you of all people know how much I'm like super adamant about the goals of bacteria and organisms being diametrically opposed.
Yes.
And so the question is always, okay, so why are the bacteria going to maintain this modification? And the simple answer is because those bees don't die.
Yes.
And so it would be a slow process, but it would be a evolutionarily viable process, if that makes sense.
Yeah.
because it's going to work better. Which is something I'm really excited about, because I like it when you can modify something and then you don't have to like spend the rest of your days like redoing it over and over and over again. Remember, I'm lazy.
Yeah. Also, anytime you've got to redo a protocol in bio, you just go a little bit more insane until by the end of it, you're just like, you know, like when it does finally work, you're just like waving streamers and putting a sparkler up your ass. It's just, shit gets weird. Like, so this is really cool. I wonder if the easiest way to test if this is bee specific would be to get like some silkworms or some other very easy to grow.
I'm putting
insects
together that zebrafish laboratory.
Well, yeah, but I think it's best to start with an insect. Is what I'm saying. I mean, we'll talk about the laboratory in a second, but like, I mean, I think silkworms could be, or some sort of moth could be a really interesting thing to start with. A, because they're easy to get. B, because nobody really cares if they're neonic resistant. Because like if you did that to a cockroach, everybody would hate you forever. But if you did it to
cockroaches are very, very important.
Not the invasive ones. And like if you do that to like a Madagascar hissing cockroach and then release it in Florida, Florida would be just as fucked as it always is.
I was going to say, there's nothing going to be different about Florida.
Yeah, nothing. It'll die to the kudzu. It'll be fine.
Speaking of Florida, I have an invasive species right now.
Oh, are we gonna introduce your invasive species?
Oh, totally.
Okay.
Hello.
This is Pistachio.
I was the hands earlier.
Yep.
So yeah, Pistachio was my intern-ish
Friend.
Yeah.
Lab partner. Pistachio worked at the lab down in Florida and helped a lot of the projects. And he came up to visit, and he's part of the reason that I had an extra pair of hands to work on bees.
Indeed.
So.
And you know, needed an excuse to escape Florida and the doom that is happening there.
Also, we tested a vaccine.
Yeah. That was fun.
I mean, price of admission.
My first stabbing.
Yeah.
It's a biohacker thing.
Some of the comments. You can't break Florida. Florida is its own breaking. I mean, in a sense, yeah. You can't grow more mold on a moldy petri dish. It just is the way that it is. Okay, so let's talk. I wonder if I should have the bee plasmid here.
I mean, it's in the folder.
Yeah, but I should have it in Benchling already, and I have Benchling open. Just as soon as it fucking loads. Also, just for those wondering, the Plasmid synth donation bar thing was for this Plasmid. Definitely not Zombie Serum version 3. This is a cancer therapy that I'm developing that is based on a malarial protein. So that's fun. Okay, let's see. Do I have a folder labeled bees? Yes, I do. Bee symbiote construct, I believe, is the right thing?
Yep.
Okay. Oh, God.
Also, again, if anybody is interested, this is the link to the bee folder.
I forgot this is an Andreas Plasmid.
Yep.
Oh, God. These are always so convoluted. Fuck.
No, they're perfectly designed.
And
which
nothing
is technically
makes sense.
wrong for biology, but they're great. They work, and that's what matters.
I fucking hope so. Well, because the thing is, like, none of this shit's labeled. I have to call him after this and give him shit for not labeling his thing properly. Because there's like these huge gaps of like, what is here? I don't know, but I know him, so I know he put something here. You know what I mean? Like,
yeah,
he may have labeled. He's like, this is feature 8. Okay, well, where does feature 8 start and where does it start? This is feature 9. Okay, what are they though? Like,
yeah,
this is feature 21. We were at feature 8. Where did the other 11 go?
Yeah,
This is what I'm talking about. Like this Plasmid, I'm sure will work. I'm sure that he has
No,
put all sorts of...
it's unfortunate. This is the tragedy of the Andreas genius is that
he's very good at building DNA, but I have to
he
give
has a
him more...
perfect model in his head.
Yeah, I just need to give him more shit
Yep.
about labeling his thing. If there's one thing that I will continue to harp on every time I do one of these streams, it's label your goddamn plasmids.
Oh my god, label everything.
Everything. It's like
constantly.
It's like commenting code, except if you don't label it, it might just not work. Like, it's not like the
or
comments...
it'll kill something.
Or it'll kill something. Like, it's not like comments are optional. They're fucking mandatory and people need to do the thing. Like, it's not that hard.
Okay, so two quick responses to some of the comments.
Okay, sure.
One, so this is, I'm just going to throw it into a bunch of various things. Comments that have popped up. One is that bees are actually like grooming animals. And so the way to distribute this would just be to like mist a bee
With sugar water.
with a sugar water that's been inoculated, and the bee is going to lick itself, and then when it sees another bee, that bee is going to lick that. So,
Or just so what I was thinking, me and Gabe talked about this a little bit before the show. You can buy, especially if you live in a country that has winter. If you're raising bees, you can buy a specific like feeder, so that if a colony is a little bit weak or a little low on food, you can basically it's a tank essentially with like a little feeding port that you fill up with sugar water so that you can just feed the bees. So in my head, that seems like if you want to just treat one or two, or like treat a hive with this, that seems like probably the best way to do it. Because then you can just like dope the feeding solution. All the bees will take a drink, and then it should affect all of them. But I mean, you could also just like if you want to treat like a lot of hives, you can just kind of go around misting them. The only thing I would say that is probably a terrible idea with the misting is if there's any varroa mites in your hive, you've now made the varroa mites neonic resistant as well.
true.
That's
Yeah,
really the
you know,
biggest
we
problem
actually
with that.
The centrifuge tube and spun the bees with the sugar. So cute.
You did what?
No, we didn't do it.
Uh,
In the paper, though, they
What?
centrifuged the bees.
Don't centrifuge the bees! What the fuck?
the
Poor
candy box
bees.
experiment.
That fucking bee is probably just sitting there going, what is going on? That's okay. That's
And then there was another really quick question about robot bees.
Oh god.
oh,
And
it's
no.
just fucking
Okay.
no.
The solution to breaking things is not to get rid of them and make more things.
Also,
It's
robot
to
bees
fix-
can't work. Like,
Yeah, it just doesn't.
The
You have
amount
no
of
idea how much pollination is going on.
that, and also just the energy requirements of a thing that flies. Like, no battery on Earth could power them. Like, the bee would be the size of my fist, and it would have a flight time of like 10 minutes. Like, it's not gonna.
Fisted by bees.
Yeah, that's a bad day.
Title of my no.
But yeah, so you can't. So it's not. So just to be really clear here, as much as it might feel like we're living in an episode of Black Mirror, we're not living in an episode of Black Mirror, and so you can't make robot bees. They would have to be like, you'd have to run them off like.
It just doesn't work that way.
it just-
Thermodynamics are a thing, people.
Well, I mean, there's one thing that could have enough energy density to power them, which is a fucking stick of plutonium. But I don't think you really want like a rod of critical plutonium flying around, like
I mean,
in the
if you're
thousands.
soft bleak, maybe.
Yeah, maybe, but like it's a bad. It not only is it a bad idea, it's thermodynamically unrealistic, mechanistically unrealistic, and also we have fucking bees. Like, it seems easier to just not kill the bees than to try and engineer some shit that's gonna do
It's
half
like
as
when
good of a
people
job.
are like, "Hey, scientists have developed an artificial leaf," and it's like, you know what else makes leaves? Trees, motherfucker.
Yeah, it's like you could make giant living solar panels, or you could just grow some fucking trees and like stick your solar cells in a desert. Like, also, I mean, that doesn't even get into the whole. I find solar extremely stupid. Like, On a small scale, it's fine, but like this whole like we're gonna save the world with solar panel and windmills. I'm like, that's not how thermodynamics works at all, or energy density, at all. Like anytime somebody's like, we need more solar and wind, it's like you're basically saying we need more gas turbines, because the only way that you can have a grid run off windmills and solar is if you also have gas turbines to pick up the slack. That's just how that works. The base load is too high, and the sun goes down, and the wind stops. So, yeah, really the only tech that works is nuclear, but we're not getting into that today. That is a topic for another day, because I can rant on that shit for hours. Okay, so this is the bee construct.
Right.
I'm gonna have to.
So somebody here is chatting about how trees don't necessarily have optimal photosynthesis. Optimization is bullshit.
Yeah.
Okay.
Optimization is the thing you do with computers.
We like to do, but that's not how biology works. Biology works with functionalization.
Yeah.
Functionalization means it does the thing. We don't want to have an optimized because optimized doesn't give you any wiggle room. It doesn't allow for play in the system. An optimized system is a system that breaks.
Yeah.
Also,
Have you ever
the previous system.
Exactly. If it was optimized, why doesn't it already exist? Because optimization doesn't work.
Right, like why are plants not black? Like if they were black, they would absorb more energy, except they would also overheat. Like
because all plants matter, Justin.
Jesus.
No.
Just don't.
Yeah. No, it's fucked up.
That's super fucked up. But anyway, so like the other thing, yeah, you ever there's an expression, and I'm probably gonna butcher this, but it's something that I've heard mostly in like movies. I don't know if I've ever actually heard an actual person who races hear this, but like don't tune an engine too tightly. So like something that might be really, really fast or like really super duper optimized, you hit one bump, it knocks something out of alignment and the whole fucking thing fails. Like this is sort of the problem with an optimized biological system. Like if you've gone in and you've tuned everything so tightly and you've removed non-essential things, all you've done is open it up to breaking. Like these like biological systems have been around for literally billions of years. And yes, they look like on this, and I mean, yes, they are basically held together by duct tape and zip ties, but it's bespoke duct tape and zip ties, and it without those redundant systems built in, shit fails very, very, very easily. This is why, like, when everyone talks about, like, human DNA is 95% junk, and it's like, well, no, we don't know what the other 95% is, and also, it's only, like, it only does nothing when you're healthy. Like, that other 95% of the DNA turns on as soon as you get sick. It's all extra stuff for dealing with things that can go wrong. So, like, when you make,
they call it dark DNA.
Yeah, now it's dark DNA, not junk. It's also just fun side note. The idea that we've actually sequenced the entire human genome is a myth. We have not. There's certain areas, like, near the centrosome, or centromere, near the chunky, chewy center of the chromosome, that is extremely repetitive. So all of our sequencing methods are utter garbage at being able to sequence this, because it's very hard to tell where the things go. But we know that if you fuck with one of the letters, lots of very important things break. So technically we've sequenced the human genome, just not some of the important bits because they're too weird. And like, this is an ongoing thing in biology. A lot of we've done a thing, I mean technically, but not accounting for the fact that biology is super fucking weird. Like, I'd say on a good day, we know less than a fraction of a percent of about how bio works. Like,
yeah, depends on the field, but generally, yeah.
And the organism even. Like, it's like, you know, we know a fair bit about mammalian cells. We don't know shit about most other eukaryotes.
One of us is clown and one of us knows. Okay, you know what? I'm not going to respond to that.
What?
Sorry, it was in all caps. It just jumped out at me. I'm going to move on. Yeah, so somebody was talking about genetic engineering is great, but it doesn't stop habitat destruction. That is absolutely correct.
It is.
Genetic engineering is a tool. Permaculture is also great, but it doesn't stop habitat destruction. So, like, all of these things are just pieces, And being able to integrate these pieces is actually how I think we're going to be able to move forward as a species, because you can't just use one tool all the time. You can't just like punch things into submission regardless of the type of tool that you have. Like if like genetic engineering is a hammer, permaculture is a multi-wrench.
So there's
multi-wrench is cool. Multi-wrench is shit at putting nails in boards.
It is. So I just saw a comment. So it says, sure, but if you're fucking around with customizing bees and eliminating cancer, you're already talking about purposeful optimization. That is not true. Optimizing it
This is a repair work.
Yeah, this is some shit is broken. We're patching a leak. It's like patching code. It's different than optimizing. We're not trying
to make them better. We're trying to fix what we've already broken.
It's like the
right.
argument against in general.
Yeah, this is the general argument against the ENTS project.
Yeah.
Also, where they're like, oh, you're just like you're trying to do this thing, and it's like, yes, well, everything else is broken.
Yeah.
And so we're trying to do something different.
Also, with the cancer thing specifically, we're not trying to optimize anything. The mod should go away after about 4 weeks. It's literally like, your system is broken. That's what cancer is. Like, some cells are so broken that they've decided that their life is worth more than yours. So all we're doing is just clearing those out. That's like saying taking chemotherapy is optimizing. It's not. It's just trying to prevent you from dying. There's a distinct difference. With the bees, it's the bees are dying. So something is broken. In this case, it's the humans fucked with them. And so all we're doing is just kind of making some slight repairs. That's all this is. It's like, if we were trying to like make the bees the size of fists and also shoot lasers and like, you know, make
dragons,
make honey hallucinogenic, then yeah, now you're trying to optimize bees, but also it's not going to fucking work. So it's a distinct difference. Okay, so how are you going to actually get the DNA into the weird bacteria?
Grab the thing. Electroporation.
Are you just going to electroporate them?
Pew pew, buddy.
Okay.
Where are the cuvettes?
Okay, so for those who aren't familiar,
this works so well. I am like constantly stunned about how effective this is, except with the clean coli.
Yeah, the clean coli fucking suck. I absolutely hate them.
We hate them.
I fucking hate them. They're so for those who are unaware, the thing that he was just brandishing is what's called an electroporator, which is basically you use essentially a bolt of lightning to force DNA into usually bacteria, but it can be other things. And so electroporation is really, really, really effective. So on all the videos that I've made on my channel where I modified bacteria or yeast or whatever else, I used a method called heat shock, where you basically heat the thing up, like you mix the organism with the DNA, you heat them up, cool them down, and that kind of stresses them out enough and makes the membrane permeable enough that the DNA can actually end up inside the cells. Whereas electroporation is you basically mix the organism with the DNA and then you shock them, and in that fraction of a second that there's like 1,000 or 2,000 volts applied to the solution, it goes from basically like the membrane of the organism rapidly becomes porous, so like little pores develop, and the charge forces the DNA through the hole. And then as soon as the charge dissipates, the pores very rapidly close, and then they're modified. And the effectiveness of electroporation is something like 10 or 100 times higher than heat shock. Heat shock is just nice because it's easy and, frankly, it's lazy. Because it's literally like, I got some hot water, fuck it, see if that goes. But electroporation does require special, they're called cuvettes. So it's basically fuck it, I'll just pull a picture. Cuvette. So this is what an electroporation cuvette looks like. It's literally a little box with two metal, like it's a little glass box with two metal plates in it. Yeah, they come usually individually packaged. They're reusable though, which is nice. If you clean them out real good and then re-sterilize them, they can be reused. The way that you set how much voltage is actually applied to the cells is the gap between the two plates. So if it's a wider gap, you're going to end up with a higher voltage than if it's much slower, just because the breakdown characteristic of the solution, basically. But yeah, so electroporation is very effective. So you're going to be using electroporation. That's very cool. I'm really interested to see how well those are going to be modified, honestly. Unfortunately, because Andreas doesn't label his shit, I can't tell what...
Hey, we talked about this.
What?
No shit talking.
Oh, no, I'm going to continue shit talking until he labels
Only
his
between
shit.
us.
No, I'm going to continue shit talking until he learns to fucking label his constructs.
Fair enough.
I
can't argue with that.
like, because the thing is, I'm looking at this and I'm like, what antibiotic are you using as selection? I have no idea. It's not labeled. Like, truly, I legitimately
there's a
have...
better one in here, though.
It's not his fault genetic engineering is banned in this country.
Yeah.
Give him a break.
Yeah, that's fair enough. Yes. Oh, so I've just quickly, there have been some comments asking for a Discord server. I do have a Discord server. It's available for patrons and channel members. I'm keeping it small and I'm keeping it to patrons because I do not feel like moderating a Discord with 10,000 people in it. So if you're a patron, you get access to the Discord. That's how it goes. In fact, I have the Discord open on this other window right here.
Ooh, is there a whole back channel of excited people?
There is, yeah. I think there's
that we don't even know about?
Yeah, there's like 70 people in there now.
Cool.
So that's been pretty cool. And also, it's been actually pretty cool. Anytime I've had an issue, I've just been like, hey, are there any machinists? And people are like, I am. And I'm like, oh, cool. And I can like immediately sort shit out. It's actually been pretty great. So yeah, if you want in on the Discord, Patreon or channel memberships are your way in. Get on it. But yeah, so looking at this construct, I have no fucking idea how he intends on getting it into the bacteria, and so I'm going to have to kick him and be like, hey, Andreas, what the hell? But I assume you're using some sort of... God, I feel like this is some, like, it's one of these things that it's like a proton ATPase or something that, like, that's probably...
You know what the problem is?
What?
Oh, gosh. Okay, no. So, you got two problems here. One is that you're not using the SnapGene viewer, because this is, SnapGene actually has some things labeled.
Are you seeing what I'm seeing? Because I'm not... If it's a gene bank...
But then all of the features, the whole thing with the features, I think these are just marks for himself, because it's really just... so one of the things that Andreas and I talked about, so you mentioned this concept of dark DNA, right?
Yeah.
And so on another project that we were working on, Andreas is like, okay, well, the first thing I will do is I'll just knock out all the introns. And I'm like, oh, no, I don't think you need to do that. And he's like, oh, well, it'll be fine. And I'm like, you don't know that. You don't know if this is, like, necessary or not. Because we don't actually understand how these big chunks of DNA work. And so it might be the most, like, highly efficient method, but it's not necessarily the most functional method.
I mean, this is also where we diverge, because I am also of the opinion that if I don't need introns, I don't include them.
But you don't know if you need introns.
Well, but I almost never start with the DNA of something. I almost always start with just the amino sequence and then just back translate, and it works seemingly every time.
This project had, I'm going to say it was like, the gene sequence was 75% intron.
Right.
And I'm like, there's absolutely no freaking way that these three coding sequences are this goddamn far apart, and all of this stuff in the middle is just useless. Especially when you consider the fact that, like, 7% of intron DNA is conserved throughout species, which means that, like, that's high. That's, like, really high.
I mean, I'll give you that, but, like, which project was this for? Was this the lactose thing? Because I remember you.
No, it was actually it's for the it was for the Ents.
Oh, okay.
So it gets even weirder because it's plant DNA.
Yeah. Okay. So then I would almost give it to you, but I would still take them out because then the chance of that being a transposable element and it fucking with your thing is actually higher. I would still.
Well,
I mean, if I was designing this, I would still remove them.
We originally started that conversation though between Andreas and I, is because we were talking about the lactose project, and I'm like, okay, so what if you do this thing, and it works, but then you're missing these important genes that get upregulated or turned on when you're sick, and like you get sick.
But I mean, with that one, like, so with the lactose thing specifically, I would have definitely removed the introns because the introns are where the regulatory elements that turn the fucking thing off are, which is exactly what you want to avoid.
Yeah, but it's also probably going to have important.
Yeah, but your DNA already would contain those. So, like, if you're doing the lactose project, you just want the gene to turn on. I don't care about the regulatory elements. They're already there. I just want the stupid protein, which has turned off, to not be off.
But wouldn't that also be a signifier of underlying activity as well? Like, if you have that innate condition, there's probably something else that you might not want to fuck with?
No, if you have that condition, it's because you've got an SNP that happens to code for you are fucked. That's it. There's somewhere between 7 to 9 SNPs in the lactose gene chunk that if you have them, the gene turns off. If you don't, it stays on. And so technically the mutation is that you've got the ones that leave it on because in most species it turns off. But if you're trying to do a gene therapy where you're trying to make a protein stay on, you definitely want to remove that. And so like this, I mean, it really, really does depend on what you're doing and how you're doing it. Like a lot of the time at least from what I've seen, a lot of the time where things like introns are left in DNA, it's usually because of the way that DNA was assembled, not because that this was a explicit choice. Like most of the time this happens is if you're cloning a gene out of something directly, rather than if you're doing it the way that we do it, where we tend to synthesize things. Because if you're synthesizing, you can just get rid of all that shit, have it just make the protein, and most of the time it works. Whereas if you're cloning things, like using PCR to isolate the gene, more often you're going to include those regulatory elements because there's literally no way to remove them. So you're like, fuck it, hopefully it doesn't break anything. Like, and
right. So the point that I was trying to get to here is I'm fairly certain, and now I just feel like he's asleep because it's in Austria right now.
Yeah. Well, wake him up from his coffin. Or no,
you're like, Andreas, what the fuck is feature 8?
What's feature 27? Fuck feature 8. I don't know what's 1 to
So these are just markers because it's all just one long gene here. What we're seeing here are him making like little tags, I guess, like footnotes of things that he's maintaining.
I mean, maybe, but
I've watched him edit. He just highlights shit.
All right.
So anywho,
I don't know. This is it's like different coding methodologies. Like he designs things very differently than I do.
It's only 10 here. Okay, I'm doing it.
Yes.
I'm doing it. We're getting Andreas in on the chat.
Oh, thank God. Like, somebody needs to kick that boy and be like, what is going on? Why do you do this to me? Oh, this is great. So I mean, just to juxtapose these things, this is something that Andreas made, right?
Nope. Hold on here, Andreas.
Oh my God, he's awake. Hey,
I'm on a live stream with Justin right now, and we are looking at your bee symbiont construct.
Oh my God, it's perfect.
And you did not comment your code well, and we're super confused. So there's things that say like feature 8, feature 24, and when you mouse over them, they just show the little chunk of code.
Or like specific names of genes, but not what they do. Which one?
What is feature 8, Andreas?
Which one? What antibiotic does this
Okay,
use?
he's pulling it up right now.
Okay, good. Ask him
Pulling it up right
ask
now.
him what antibiotic
Hey,
it
this
uses.
is a party. We've got a Pistachio, we got an Andreas, we got a Justin, we got a Gabriel.
Yeah, can you?
Yeah, Pistachio's here.
The whole game.
Yeah.
Andreas!
Okay.
Ask him what fucking antibiotic this thing uses, and where. Which feature is it? I think it's this one because it says
Okay.
apotheosis
Hey,
inhibitor.
Justin. Shush. Okay, so we see two different promoters pointing in two different directions, correct?
Okay. Oh my god.
Mm-hmm. Okay, so they both yep. So hey, I gotta translate. So they both transcribe the gene from different directions so that we get double-stranded RNA. Right. So sense and antisense. Okay, but what are these features, dude?
Ask him about the antibiotic.
Oh God. Okay, so what happened? What happened is that he basically got the sequence from a really shitty source because nobody else has the thing available, and so he got like some garbage, and so he basically used BLAST to confirm that this thing was the thing. So all of those markers are basically chunks that he did in BLAST to make sure that the thing that he had was the thing. So like I said, they're markers. They're not actually like extra features. Justin wants to know about the antibiotics though.
Please don't tell me he didn't add one.
Well, because we're getting it synthesized, it's going into a backbone that has an antibiotic. And so it doesn't actually
Why
have...
is that not in the fucking file?
Because this doesn't have the backbone. This is just the construct. This is just the print.
Then this file is useless. Where the fuck?
No, this makes a lot of sense.
I mean, in a not at all kind of way, why wouldn't the file include the backbone? Everything I've ever designed always includes the backbone.
This is just for the printies.
Just a general backbone, any backbone.
Yeah.
You don't just choose any backbone. You got to choose specific backbone.
Not for this particular situation. I think it went into is it going into a puck? Oh, B bacteria. Yeah. Yeah. We kind of went into a library and found a broad range backbone because it won't work in PUC. And then but we're using the antibiotic that the backbone has. It's not in the construct. So looking at this thing as a ring is actually an incorrect way of looking at it. So thank you, Andreas. I'm going to let you go to bed now. No, I'm on a camera. We'll talk about this later.
Oh
okay, bye.
This whole fucking thing.
No, so the way that you're looking at it right here, the way that you have it pulled up is actually incorrect because it's not. This part is not supposed to be a ring.
So this is okay. I mean that I can change.
It's a linear piece.
Fine, it's linear. Update.
And so and then it gets smacked into a backbone.
Okay. It's still weird that I don't have a file that includes the backbone. Like everything I've ever designed. like this, for example.
It's just a piece of things.
Contains backbone. Backbone very important because it contains all of that missing information that was very frustrating. Like,
And so yes, the all of these other, so everything that says feature
right?
is just like him documenting and fact checking that the construct was a real thing.
Okay.
That's it.
So this is literally just one fragment taken directly from some shitty paper.
Uh-huh.
Oh, okay. I mean, I am actually more satisfied with that than the alternative, because the alternative was like deeply frustrating and like,
Well,
Yeah.
it'd be like that sometimes.
Sometimes it be like that,
Thank
as
you.
the kids say. Yeah.
okay, so I've removed all of the feature notes because that shit's just annoying. So now, I mean, this actually, so basically he has gone through, so this is the weird Varroa. This makes me think he took it out of, that this was taken out of Varroa mites. Also, what was the deal with the double sense RNA? The fuck is that?
What? So he can have double-stranded RNA.
Right. Why would you want that? I can think of no context other than like antiviral shit that's ever helpful.
And why are we doing this, Justin?
Antibiotic resistance, which should be one protein.
No, this is just how it works. I don't know why. There's like one paper about this.
Okay.
Their explanation of mechanisms is fairly bullshit, in as much as that it's not very good. But they do have a good proof of concept, and so we're working off of that.
Okay.
Yeah,
Not.
it's still really cool.
It is still really cool. I'm just progressively more skeptical of it working. Like, okay.
Hey, you were also skeptical of me being able to isolate these bacteria.
And I was pleasantly surprised when I was wrong.
Yep.
Just that is like
Now,
putting
that's
some
what I'm here for, buddy. Pleasantly surprising you when you're wrong.
This is just, God.
Let's be honest. It's because I was here.
It's because Pistachio is here.
I'm gonna just go ahead and blame Pistachio, because
I know, this is my lucky high school kid.
Yeah. Made the thing
it's
work.
It's much easier to just blame Pistachio.
Usually we blame Andreas. He's not even here.
I mean, I still blame
so somebody
Andreas.
just said, I don't know why, and said this literally never happens in any other science. Oh my god!
Yes, it fucking does.
Okay, computer science is just engineering. Engineering is definitely engineering. And every respectable, reasonable scientist, if they are not capable of saying, I don't know how this works, they are lying to you.
Yeah,
and themselves.
Dude, physics. If any physicist ever tells you they know how quantum mechanics works, just, like, slap them upside the head, because they're clearly lying to you. Like,
Also, distraction?
we can't even decide whether locality is real. Like, we can't, like, that's as basic as shit gets. Like, either realism is real or locality is real, but it can't be both. Like, that's sort of underpinning of all physics. Like, so yeah, no, anyone who tells them, it says that they know exactly how something works, is probably full of shit. Like, I mean, there are exceptions, but even the exceptions are generally, well, generalizations. Like, like in chemistry, you know, we draw all these like really clever diagrams, like, and look, the electrons move around in this way. Do they though? I mean, like, maybe, probably, but like, we're talking about a quantum mechanics thing. The electron can be on the
For
other side
those
of the
who are
fucking
curious
universe.
about why he's named Pistachio, by the way, I've seen this pop up.
He named himself that.
I named him that.
Oh, okay.
Yeah, no, I'm really shitty with remembering people's names. And so after 2 weeks of calling him high school kid, I was told that I have to give him a name. And instead of using his name, which is Alex, by the way,
Hello again.
I just started calling him Pistachio, and it stuck.
Yeah.
Yes, that's how it worked.
Yeah, that's the thing. Also, I'm seeing a comment here. Quantum mechanics is extremely predictive. Then you have not looked at quantum chromodynamics, have you? None of our predictions make sense or work very well. I could list off so many things in quantum mechanics that we don't understand why they're like that or how they just absolutely throw a wrench into what we know. The only thing in quantum mechanics that's extremely predictive is quantum electrodynamics. And even then, shit is so weird and requires like massive supercomputers to just like churn through the bullshit.
We'll just keep going forever, though.
Somebody asks, which does Pistachio prefer? I mean, Gabe gets to call me Pistachio because I know it's not a derogatory term. So I'll take it from my close friends. You can call me Pistachio or Alex if you want, though. I don't care.
Definitely Pistachio.
It's my bio name.
He has resigned himself to being a decorative tree nut.
Yeah, it's better than being a turnip.
Ooh. Hello,
Whatever.
Turnip Garden.
Whatever, I own that shit. I like a nice turnip soup. Whatever. Yeah. Anyway, so now we're just getting lots of comments about how quantum mechanics is dumb, and I agree. But anyways, so we have solved.
Do we have any more questions about bees?
What do they taste like? No. There's a.
Sasha, give me the bee leftovers.
Oh God, no! Do not eat the bees.
It smells so terrible.
Oh God.
The isolation.
So I did a video about plasma coating things in metal, and I had a jar of hornets, and they were.
Oh, that's right.
Yeah, they were
so cool.
They were real fresh when I got them, and then they sat in a jar in the lab for like months, and they just got progressively more disgusting, and eventually we had to throw them out. But like, yeah, rotting bees and/or hornets is not a good smell. Can confirm.
Yeah, I have to not breathe whenever I'm getting.
The term is fermented, Justin.
I think they're just rotted at that point, honestly. There's a subreddit r/bee eaters.
I did not.
Yeah, it's one of my favorite joke subreddits.
So in theory, so somebody asked a question, an actual question about bees, as opposed to us being weird. Somebody asked, will this work on all bee species or species?
I was about to say species.
Because there are over 20,000 types
of bees.
The answer to this is it should, in as much as that this is a highly conserved gut bacteria across multiple species, and it is, as you saw from the previous pictures that Justin pulled up, kind of super prevalent. So hopefully, we don't know.
The hardest part was getting
yeah, the hardest part so far has actually been catching the bee.
Did you just catch a wild bee?
Yes.
Yeah?
Oh, for fuck's sake.
I thought you get your bee. I got a pistachio. I gave him a jar. Sent him off into the wild.
And you're sure it was a bee, not just like...
Definitely a bee.
Definitely a bee? Okay.
Definitely a bee.
There's just one beehive near you that's like, where did Gary go?
Kind of, probably. Yep, and then we ISO'd the fuck out of him. So
we know it's not from the outside of him.
Oh, okay.
Yeah.
Noted. Well, it'd be a lady, but whatever. So this sounds like you're gonna need to buy a beehive. This sounds like you're gonna need to get some bees.
Or we could just make friends with a local person who has an apiary.
I mean, sure, but then you could own bees.
I know. I'm still working on trying to get chickens right now. So
that's true. Oh, so Vesta has a beehive. So you should.
Oh, cool.
Yeah, you should definitely send me some of your weird modified bacteria once this is done.
Yeah, absolutely.
We'll
and yes, we
modify
are
her bees.
totally going to test this.
Okay, cool.
Doing research without follow through is kind of just wanking off all the time.
Yeah. Also, so there's a question here. Can we make bees produce stuff to fight off the varroa mites, like a specific mite pesticide? I mean, that's kind of what we're doing. Like, if these are...
Talk about the... Well, so neonics actually make them more susceptible to the mites, but then there's also, like, stamets stuff, right?
Yeah. I mean,
so
I like the...
mushrooms.
Yeah, it's the mushroom feeding and all that kind of stuff. That's actually really cool. But, I mean, you could make... I wonder if you could make BTBs.
I'm sorry, what did you just say?
BTBs. Like BT producing bees.
Ah, okay. I'm like
BTBs.
BTBs.
BTBs. It's the more fun version of diabetes.
That's pretty good.
um, hey, I got a solid snort laugh. I'll take that as a win. Um, but yeah, so I mean, if you could maybe make BT producing bees, if you could also make them resistant to BT. Um, but I mean,
Well,
that could
okay,
have
so
all
this,
kinds
Like
of
the
weird
malaria
shit.
mosquito thing.
yeah, this
Yeah.
and this also gets into the like one of the things that some people were talking about a little bit, and definitely something that I'm always ready to talk about, which is you can't engineer a perfect solution. And so like this starts to turn into okay, you need to start creating a more robust environment that supports the bees because you just can't keep cramming gene mods into everything.
Yes.
Genetic engineering by itself will not work. Um, it we have seen this. It just doesn't. Um, and super hardcore, I'm going back to nature also does not work. It's just not the world we live in anymore.
Yeah.
So there needs to be a combination of these two things. So
Also, there's like mechanical methods to get rid of varroas. So there's this thing called that actually Vesta has called a solar hive. So it's basically like this cell that you put above the beehive, that's kind of like a tiny greenhouse. So I believe I don't remember the specifics of it, but it's something along the lines of there's some sort of mesh that basically lets the mites get in, but not the bees, or maybe the bees get in and not the I don't fucking know. The
Also,
point
the
is,
mites would only be a concern after we have a significantly increased population of bees, right?
like in a
Because in
hive.
theory, things would kind of balance out if there's
No, they breed very fast and they can like dick a hive really really quickly. But so this solar hive thing is basically like essentially a mechanical method to get rid of varroa mites, and apparently it works pretty well. So
Mr. Condi has, I don't know if I'm pronouncing that right. I don't know. It's the screen name. Has a question about like testing and then there being like an escape bee that gets the bad genes out into the wild. It's like if they're bad genes, then they don't work.
yeah.
So the concept that they're going to take a thing that doesn't work in an environment that is all about things working and then like somehow just like make other stuff not work doesn't make any sense. That's
Also let's be really clear here. If all of a sudden everything was resistant to neonicotinoids, that means we'd stop using them. That sounds pretty good to me, honestly.
Yeah.
I see this. I see this as an absolute win, frankly.
This is a win-win.
So bioengineering is like enchanting. I mean, kind of. You know, we need Eye of Newt and you know, a collection of. I had to use salmon sperm in my last thing. That was weird. I have like a freezer full of salmon sperm. Biology's weird. Get used to it. Like Gabe has a freezer.
Stop masturbating fish, Justin.
I don't masturbate fish. I pay other people to masturbate fish for me.
Fair enough.
Because I'm a professional. I mean, like, I was actually so somebody commented on the well, actually a lot of people commented on the yeast video about the salmon sperm thing, but I was like, the fact that I had like frozen peas and salmon sperm in my freezer for doing this protocol is like some of the least weird things that any of my bio friends have in their fridge. Like,
yeah.
like I mean, you have like cases of like baby cow blood and also blended up bee and also
I want to be honest here. It's not baby cow blood. It is the blood taken from the fetus while it's still inside the mother. Okay, if I get to be a witch, I want to be super fucking awesome about this.
Okay, fair. Okay, so it's baby fetus blood, which is somehow worse. But I mean, like
scientists are monsters.
Basically, okay. So on the list of other insane things that my biologist friends have had in their fridges, I had one friend who had an actual wolf head that was slowly rotting, which she collected in BC and had to drive all the way to Saskatchewan before she could process it. So she just had this like cooler full of slowly rotting wolf head. We've had you've got your baby fetus cow blood. We've had I mean, like, Sebastian has, like, a whole tube of the screams of dying grass.
Yeah, that's true.
Which is, that one's fun. Like,
And then everything related to fermentation.
can I just say that I wish they gave things more interesting names. Like, instead of calling it, like, what's the name of that acetylstrychnone or whatever, the cow screams, or not cow screams, the grass screams. Like, I wish they just called it, like, grass screams. Like, that would make reading protocols way funnier. Like, instead of being like, add 20 micrograms of acetylstrychnone to the solution, be like, add 20 cc's of screams of grass.
I mean, this has been one of the...
And say the enchantment and sacrifice the goat, and then your protocol works.
Yeah.
It's also, like, we've talked about this before, but, like, I think biologists are some of the most superstitious people I've ever met, whilst simultaneously being the least. Like, because it's like, if you told me that, like, I have to sacrifice a goat and, like, light a candle and say a prayer and that'll make my protocol work, I'll fucking do it. Like,
sometimes we just don't look at the cells.
Right?
Yeah,
like,
that's the thing.
I'll
Do some work. I'll do some work, and then I'll just be like, just put it in the incubator.
We don't look at it for 2
So
days.
Sasha's like, do you want to look at it? And I'm like, it's not going to change anything.
like,
Like,
it's like, don't look at the groundhog. If the groundhog sees its shadow,
Schrodinger's mammalian cell.
right? It's like, if you don't look at it, you can assume it's not dead yet. Like,
Exactly.
and then you look at it and it's dead, and then you're sad.
Go get one of those bad bottles. It's on the shelf that we made.
Oh god. That one was fun. Yeah, somebody's like, don't forget the mandrakes. Yeah, mandrake is actually a very interesting plant with a whole bunch of very interesting compounds in it. And yes, it'll definitely kill you if you eat it, but it'll also get you super high once. It's in the category of things you can eat once. Like, it's like all mushrooms
Go
are...
with the good one. Oh my gosh, it's swelling. It's going to explode.
God.
We have to get rid of
Wait,
these
is that
soon.
yellow? Is that DMEM or is that FBS?
Hang on.
Oh god.
Boop. Boop.
Oh no.
So this is DMEM mixed with FBS and pen strep. We use it for growing mammalian cells. It's got phenol red in it that allows it to show you when something is like super, super wrong. Because like the things that are alive in it, notice there's no changing because this is my sterile shit and there's nothing alive in it. It just stays like Kool-Aid Man.
Yeah. It's science Kool-Aid.
So I ordered a new batch.
Is that the DMEM or the FBS?
This is DMEM.
Oh Jesus!
They were all like that.
It's sealed. It's
God.
sealed. And notice how wonderfully... Oh, yeah, let's just stir that up.
Okay,
oh,
so
yeah.
just for context here, when you're growing mammalian cells, the phenol red will change colors based on how your cells are doing. So if it's like a distinct like pinky purple, it's been out of the incubator for too long. If it's like a nice orangey red, then it means that everything's doing good. If it turns yellow, you fucking panic. Like, yellow is really, really bad. Because that means it's gone acidic.
It arrived this a little redder than this.
We are not opening it.
Yeah, it's
actually the bottle has... I've let it sit just to see how messed up it is. And the bottle is starting to swell, and you can tell that there's a distinct... Well, I can't see through it, which I should be able to do.
Yeah,
even though
it's
this thing is
clear.
sealed, it showed up contaminated with bacteria.
Which is bad. Also, that came from thermo. Like, that's not supposed to happen. Ever.
I got an instant...
I mean, yeah, to their credit, they did refund you.
And then they sent me a batch for free, so thank you.
Yeah, see, Icarus knows what's up. Yellow chem bad.
Yeah, yellow. Yeah.
Yellow chem bad. It's true in biology too. Yellow biology, very, very bad.
Yeah.
Electrolytes.
Yeah, the fact that it's opaque and yellow is horrific. Like, I've seen a lot of DMEM. It's supposed to be crystal clear. Like, there should be no sediment. There should be no nothing. It should just be clear and red.
Yeah.
The fact that that's sealed and opaque and yellow is truly
I'm like, how do they do that?
horrific. That's
yeah,
like...
No. This was the beginning of, like, a really bad week of work.
Yeah. Somebody's like, just filter sterilize it. It'll be fine. No, it will not. Like, Um, well, thank you, Nico. He just kicked in a donation. Just say yellow chem is bad. Can confirm.
Yep.
So I assume you guys aren't familiar where this is from. This is from a fantastic YouTube channel called Explosions and Fire. Who his whole thing is he does really cool chemistry, but it's all about explosives. So, but he's also come to be, like us, very superstitious that anything yellow is bad because it almost always means shit fails.
Except for your bread, evidently.
Except for my bread. My bread was remarkably yellow and really good. So that's the one exception to yellow bad. In this case, well, actually, it was kind of yellow bad because it was supposed to be orange. So yellow is bad consistently. Well, so there's explosions and fire, and there's extractions and ire. So he's got two channels. One of them is actually like getting into the details, and one of them is more just like entertainment. Personally, I love both channels. They're very entertaining. And he's just done a bunch of really cool chemistry. Like he made, there's this explosive called azide. It's got 14 nitrogens in it and 3 carbons. It is gnarly. Like, but yeah, so shout out to Explosions and Fire. He does cool shit.
Cool. I have found a question to rant about.
Oh God. Okay, go for it.
Mr. Condi, I'm loving your input. So, if that gene mod makes it into other things, it's going to force us to do something different. And while we are concerned about the damage to our crops and everything, people who do engage in permaculture agriculture don't need to use these things and somehow manage to grow food. And on top of that, you can consider the fact that 70% of the food that we grow in the United States never makes it to our table.
Yep.
And so
for like a variety of reasons.
We're concerned about the lack of crops. What we're actually saying here, what you're saying, whether you know it or not, is that you are concerned about the maintaining of a certain type of super wasteful industrial agriculture.
Yeah.
And so I don't care.
Yeah. Also, so the other thing is, like, I've been helping a friend of mine pen a letter to that we're trying to get it published in a newspaper and send it to MPs and stuff. But one of the things that me and him talk about a lot is moving crop production as some of it, not necessarily all of it, but some of it into cities, because a lot of where that wastage happens is just transport. Shit rots as soon as you take it out of a field, it immediately starts going bad. So just from the time of harvest to get it to the grocery store, we've already lost, it's like 30 or 40% of the crops. And then that other 30 odd percent is wastage from the grocery stores itself. Like, they'll throw out anything that doesn't look nice. They just won't
gotta
buy
have
it.
those perfect looking potatoes.
Which is so stupid.
And
ugly vegetables are delicious.
Yeah. And on top of that, you need to maintain the concept of non-scarcity.
Yeah.
Because, you go to a store, there's 400 freaking apples there. You know those apples aren't all going to get bought. But we have them there because it makes you feel safe.
Yes.
Also, the toilet paper. Evidently, Justin can't find Q-tips right now.
Yeah, that's weird. But so, I mean, that's the other thing. And also, like, when food has passed the point of being sellable, it hasn't gone bad yet. The grocery stores just throw it out. And then they lock their dumpsters because, God forbid, anybody get any of their food without paying for it. Like, so there's so many places in the system that need to be radically overhauled. And just being like, well, we just need more neonicotinoids, and if we get rid of the neonics, then we're going to lose all of this crop yield. It's not true. Like, the export of various crops, like, there's huge tracts of land that are basically just designated to shit we're going to export for no fucking reason. Like, so
on top of so the question of how does this like concept of gene mod gone wrong, which in all honesty, there's not a lot of evidence for that type of thing happening very much.
There is if you do it traditionally. Like, the traditional breeding has ended with
traditional breeding is terrible.
Yeah,
pugs.
Yeah, pugs. The fact that most dogs have genetic diseases
lead to less wastefulness. our wastefulness is born out of passivity. It's born out of a luxury of being able to destroy things.
Yeah.
And so if it's harder to break things, we might have to change. We have to change our habits. And in a perfect world, we are all going to just get better. We're going to become better people, and we're going to do it without any sort of strife or conflict or anything else.
Yeah, but we're living in the fucking
Yeah,
apocalypse, and that's
totally
evidently
not
not happening. We can't get people
So,
to wear a fucking
I'm not
mask, not to
necessarily
mention
saying
like
that
not
this
be
is going to
wasteful
be
bastards.
a mod gone wrong, because that's pretty fucking unrealistic. So this is kind of more playing into just like the standard GMO scare tactics type of thing, and I'm not a big fan of that. But if something did change, then we would have to change. And given that we've basically kicked the shit out of the entire ecosystem, I don't see a big problem with that.
Yeah. Honestly, there's so many little things that we're basically just pretend. It's like, it's the this is fine dog. Like, there's so many of these things. Like, a lot of our farming and the farming that happens in the like middle United States. There's two ways that we water the crops that exist there. One is through draining the aquifers, which, given another like 20, 30 years, those fuckers are going to be dry, and it takes like 6,000 years to refill them. So if there's no aquifer, you can't farm.
Yeah, I don't have 6,000 years.
Right, and then the other way that we get the water to grow our crops is actually through the Amazon rainforest. So there's this misconception that the Amazon is the lungs of the planet. It does produce an enormous amount of oxygen, but that's not actually super accurate. What the Amazon does do is produce all of our fucking rain. And not all, the rain in North America. So the evaporation from trees in the Amazon go to watering both the trees and the Amazon, so it's a self-sustaining cycle, but all the extra water ends up drifting north and is where most of the rain comes from that waters a lot of our crops. So if we got rid of the Amazon, all the fucking rain goes away, which is why it is so distressing that the fuckheads that are currently burning it down for the sake of short-term profit are like, this is why so many people are angry. Like, it's a biological resource we'll never get back. They're doing it for fucking soy and cows, which fuck soy and cows. Also, the gains that they're going to get are going to be extremely short-term. They're all going to go bankrupt, and then all they've done is they made a small amount of money up front, lost the rainforest, and then all go bankrupt. So they're right back to where the fuck they are anyway. So that gets into, like, socioeconomic, political bullshit. Not going to get in there. But the point is, if there's no Amazon and there's no aquifers, our fucking crops aren't going to grow. And, like, last I checked, that's a problem. And because then as soon as that happens, then you've got to divert huge resources to desalination. Because if there's no clean water, that's the only place you can get it. And desalination takes enormous amounts of energy. The only way to do desalination, realistically, is a nuclear plant. But nobody wants nuclear plants. So, like, it's basically like we have.
You said you weren't going to talk about nuclear stuff.
I'm not. I'm not going to get into the specifics of it. I am going to say that there are solutions to these problems. Stop being wasteful. Move crop growth into like aquaponics and local like vertical farming in cities. Like, do all these different things. Have desalination plants. Use nuclear. Get rid of all the coal plants.
How do you feel about these babies, though, Justin?
What are those? Don't tell me those are fucking bamboo seeds.
You betcha, baby.
Oh, no.
Not just bamboo. It's giant bamboo.
Oh, Jesus.
And not just giant bamboo.
So for those of you, because most of you don't know this, Gabe is supposed to be banned from buying bamboo because he has a bad habit of, how did this bamboo get here? And then everything becomes bamboo. He has a bit of a bamboo fetish, and now we don't kink shame on this channel, but we do game shame from buying bamboo
with thorns.
Oh, Jesus!
Giant thorny bamboo. Nightmare bamboo.
Oh God! What's the species? I'll look it up.
Let me pull that up for you.
Oh no!
Bambusa bamboo.
It's Bambusa bambos. It's the most bamboo ever.
How do you spell it? Bambusa Oh god.
Bambos.
Yeah.
Bambusa
Oh
bambos.
god, it's so thorny. Oh. Oh god, why would you buy this? Oh Jesus.
Because it's like the bamboo from Kung Fu films, but also with giant spikes on it.
And it's starting to germinate.
And we are germinating, buddy.
What part of you're not supposed to buy bamboo were you not understanding?
All of it.
God damn it, Gabe. One fucking job. One fucking job. Don't buy bamboo.
Yeah.
If that was my only job, I would be able to do that.
That's at least one of your jobs, and you can't seem to. So. Yeah. So for those who can't tell, because the image is a little blurry, the spikes are like multiple inches long. They're quite pokey. Why are all the? Yeah, wow, that's real pokey, all right. That's
Yeah. Pistachio just thought it would be really important that you knew that bamboo is happening.
God damn it, Pistachio! Don't encourage him.
But it's
Oh no, it's way too late for that.
you've gone too far, Justin.
When he's not looking, just put like a lot of salt in that dish, and also blend those seeds.
He's the one that planted them.
I'm also excited about the bamboo.
Yeah.
God damn it, Florida! For fuck's sake.
Yeah. There's actually bamboo. My yard is like half bamboo. In
Right,
Florida.
because Florida is
Yeah.
all invasive species and needs to be underwater sooner rather than later. God damn it. Okay, so let's just. I think we're kind of getting to the.
We're getting to the end here. I think we've just been
Yeah.
rambling.
Which,
We're talking about.
I mean, we're getting
bamboo.
good viewage, so I don't think it really matters. But like, all right. So we've talked about your plans with the bees. We've talked about your progress with the bees. So really, the next update to the bee project. So the construct, I assume, has been submitted.
Yeah.
Okay.
The construct has been submitted. Probably going to take about a month to print.
Okay.
And then we're gonna smack that baby in there. And if my super magic functional boy doesn't work, I'll borrow a professional device from Notre Dame. Yeah. Also, like we got like PEI.
Yeah.
A lot of options here.
All sorts of options.
Yeah.
So you're gonna modify the bacteria, and then so first you're gonna get the bacteria tested, make sure they're the right thing. Then you're gonna modify the bacteria once the construct arrives, and then presumably you're gonna feed some bees.
Yeah.
And then presumably expose them to neonicotinoids, which means you're gonna need to get some neonics.
Yeah, that's not a hard though.
Yeah, I know it's not hard.
Hard part will be centrifuge.
I mean, you're in the States. You just walk to
No,
Home
it
Depot. You just like walk to Home Depot, go to the poison aisle.
Yeah. Basically.
Yeah. All right. Cool. So probably a month to 2 months to get the construct made, then probably 2 to 3 months worth of fucking with it. So we'll probably circle back to this topic in a couple of months.
Yeah, a couple of months.
We'll check.
I
back
mean,
in.
I don't feel like we're gonna be going anywhere.
Yeah. Right. Like. The extent of me going anywhere is if I can finally find another apartment. That's it. And
Yeah.
that's just so I can be in one location somewhere else. Like, so there's been some questions about how we can watch your progress on the bee project, and I think we've talked about this before, but you're not really great when it comes to sharing progress updates. You kind of.
I'm great at sharing updates. I share updates when they are relevant. I unfortunately am not a social media-ish type of person, so
Yeah, that's my job.
the 15 times that I fail on something, you're not going to get like a daily, hey, this isn't working.
Everything's
Yeah,
up.
I mean, like,
But you will hear about me talk about things, and as much as Justin hates it, I make cute little videos on TikTok where I do science.
gross. And I mean, if you're on
Okay,
the,
YouTube influencer.
it pays. and doesn't send my data directly to the Chinese government. That sounds fine to me. It just
Or do you really think that this is not getting sent directly to the Chinese government? And does it even matter?
It's getting sent directly to Google,
Not
and then if the Google robot's in a good mood, I get paid. It's very different.
Remember that whole thing where
I know who my god is. My god is the robot that runs Google. It's fine. I'm used to it.
Yeah. But you
whatever.
remember that part where I don't care about money?
Yeah.
Yeah.
But so yeah, if you're on the patron or member Discord, then you'll probably see me posting about this as updates come in. Like if you want to see like failure, that's really where those updates get posted, because I also don't tend to post my failures just because they're boring, and I mean they're annoying to me, and like so really the only place I've been posting them is like on the Discord, so that I can like ask for help for stuff. So yeah. So yeah, if you're a patron or channel member, there's a link to the Discord. You can do that. There's links to Gabe's stuff in the description. I believe everything other than his horrifying TikTok.
They're cute.
I refuse.
It's cute science.
I refuse to participate.
Cute science.
Me, because it's on TikTok. But other than that, I think we're probably gonna wrap it up for the day. If there's any last questions, we'll give it a minute or two just to see if anyone's got anything that anything pressing.
Final question.
Otherwise, we're gonna wrap it up for the day. So yeah, if you got anything, that's now is the time.
No, you cannot inject plants with sugar.
Yeah. So the question was, could plants have their metabolism run by injecting them with sugar? No. just straight up no.
It's true. I am an insufferable asshole.
Yeah.
This
is true.
The alternative is that you get walked over by other people. I'm just trying to do stuff. It's not really about,
Yeah.
I'm not here to make you happy. Sorry.
It's great when he's talking to Comcast.
Yeah, I'm really useful if you need someone to talk to Comcast.
Can you feed plants sugar through their roots? Only if you want them to rot. That works very well in tissue culture, not so much when it's in a pot. Just fucking give the plants light. They'll make their own fucking sugars. I don't know. Why are you trying to, like, do this the hardest way possible? I don't get it. What?
Sorry, we're just keeping up with the chat stream. You go on.
Are they calling me the insufferable asshole?
Yeah.
Whatever.
You know what? Let's just talk about plant roots.
I mean, I am an insufferable asshole.
Okay, can you gene mod already grown trees or seeds?
Kind of.
Boogers? Ooh, grab the ends too. No. It's right over there. I put it there because I cracked the top. Yeah. So we are actually.
Yeah, go for it.
We are actually working on this. So one of the things that I really want to be able to accomplish is to make the genetic modification of plants so freaking stupid easy that you can do it the way that you make like mac and cheese, just like super easy. And so what we're doing is we're actually germinating these seeds in a mixture of what's called Scoob medium. The Scoob.
We just call it MS medium usually.
MS medium. Well, but if you're doing bacteria work, there's another MS, so I'm calling it Scoob. The Scoob media that also has our genetically modified agrobacterium, and these are basil seeds, so they're going to germinate in 2 days, and this means that they're going to sprout and instantly get infected. And so since they have such a small amount of cells exposed, hopefully we're not going to have chimeric issues, and the modification will take over the entire plant. Having a plant that's already made, like you got a tomato plant and it's this big, that's just not possible, in as much as that you're going to kind of have what's called chimericism issues, where certain cells are going to get modified and certain cells aren't, and then they're going to try to fight for dominance, and then the plant dies.
Neat.
Also, people keep on asking ENT-related questions. Watch the ENT stuffs.
Okay, ants-related question.
Where did you see ants
It's
related?
almost like, is there a way to make these grow faster?
Is there a way to make things grow faster?
model.
That's the ants project. That's what I'm talking about. That's the modification.
Yeah.
Please go back and look at previous live streams where we basically talked about this for 2 hours.
Yep, basically. I saw one question. It's long past, so I can't see who posted it. But basically asking if computer simulations would help our work in any way. No, not really. No.
You
Can't code your way out of this one.
You can't.
This is not a code problem.
Ask a question that you don't know the language for. I don't know how to say this in the most simple form.
Biology's complicated.
Actually, that's basically it. Yeah.
If you're writing code, you have to write a code to ask a question.
Yeah.
And
or
if you're
figure out
trying to ask
a specific answer.
question, it means that you already understand the system.
Yeah.
And we don't. That's why we do research.
Would need a working model of whatever it is you're trying to modify, and we don't know enough about the interactions for that to do the thing.
Right.
properly.
So it's like if you're trying to change one gene, you're interacting with literally thousands of other ones, and the environment, and the growth media, and the organism, and the other organisms that are nearby. There's so many things. You'd need a supercomputer that's basically the equivalent to all of the Bitcoin mining on Earth just to crunch through one of these problems. It's not feasible.
True story.
Just do the experiment.
The TikTok is my name, Gabriel Licina. I know Justin won't share it, but that's what it is.
I mean, it's soon going to be shut down in your country, so it's not going to matter.
That's not.
Yeah, right. Remember when Borelli thought that they were going to defund all the health programs?
Well, no, they just made
They
an
just
announcement.
fired everyone functional so that we could catch the plague.
True.
Yeah.
Okay. I
The
think.
questions about the direct seed modification for the ones in the tube, we didn't actually do any sterilization on them. We just plonked them in.
Yeah, I'm going for the laziest method possible. Agrobacterium are really aggressive, and I'm not really concerned about that.
Yeah.
On the other hand, what you could do is give it an isopropyl wash, and then give it a bleach wash, and then give it a saline wash.
Which we tried and was terrible.
Yeah, that didn't work so well.
Yeah, I mean, the standard way to sterilize seeds is you basically soak them in a 5% bleach solution for like 5 to 10 minutes, and then you just rinse them off in distilled water, or like distilled sterile water, and then you're just good to go. It's like sterilizing seeds is actually quite easy. Okay, I think we're probably gonna wrap it up for the day.
Oh, no, wait.
What? Oh, is there
The
another one?
ultimate question. What are our day jobs at the moment?
I mean, this is my day job.
Likewise,
Yeah, Gabe.
I run a genetic engineering lab.
I'm just a humble stash.
Yeah, he doesn't.
Yeah, Pistachio is a decorative
He's a
tree
student.
nut. So yeah, I mean, Gabe runs a genetic engineering lab. I run a YouTube channel and also a genetic engineering lab. That's it. That's the whole thing. We don't have that. Those are our day jobs. Like, in case it isn't clear, even though I've answered this question approximately 1,000 times, YouTube is my entire job. That's all I do. Like, it's making videos and doing research. But the research I'm able to do is because I make videos. Like, making videos pays for research, pays for reagents, pays for food, and then I keep doing research, and then I make it into videos. That's just how it goes. Whereas, Gabe has his own ways of, getting contracts and getting business, and I mean, he runs a business. Like, he does business things. Like, that's it. Those are our day jobs. Like,
I do weird things and I do them well, and then strange people come out of the woodwork and are like,
can
"Would
we give
like
you
some
money? Yeah.
That is literally my job. I don't know how it works.
It is very weird. I've lived with Gabe for a while. Like, the number of times that he just knock on my door, he's like, "Oh, so yeah, this guy offered us like this giant pile of money to do this." I'm like, "What? What do you mean? Where did they come from?" "I don't know. Came out of the woodwork." Okay. Like, this is just how Gabe do. So we just a quick question. How do you plan to combat burnout if it happens? I mean, it's ever present, man. Like doing these streams, that's how I combat burnout. Like this is fun. Like I've had a lot of comments of people being like, these streams are too long, or I missed the regular content, or whatever. These streams aren't for you. They're for my own sanity. I like doing this. It's fun. I like the interactive element. I like being able to just shoot the shit and talk about things.
It's not my mom's house. It's my house.
Yeah, Gabe owns his house. Fuck off.
Yeah.
Yeah. Also, people keep on asking about how to become a student of Gabe's, and I think that question's really weird because, like, I kind of just showed up and started doing stuff.
You need to get into the camera, Stassi.
I kind of just showed up in Florida and started being useful, or being like a lab hand. And it wasn't. I like tracked him down on the internet strangely, and then just kind of happened to start doing the stuff. Showed up every day, and then I don't know.
Yeah,
became
basically
friends. Yeah, it's not like you fill out an application or something and then.
Yeah, I mean,
I don't know. I feel like those questions are weird. And everyone's always like, "Well, you're so lucky to be working with the Gabes," and it's like, yeah, you kind of got to show up for that to happen.
Yeah. I mean, I get emails asking if there's like internships available, and my answer is always no. I'm not looking for interns. Like Gabe is looking for interns.
Lab work requires extra hands.
Right. Whereas, I mean, for me, I have very specific needs, but most of it I can do myself. Like, and the external help that I need, I've already found. So, like, and I don't have a place where, like, I could even have an intern yet. Maybe in, like, a year or two when I've got if I've got a big lab space and, like, an actual place to do a thing, then yeah, maybe. But, like, also, the idea that, like, people are going to, like, fly in from, like, London to come be an intern, like, fucking no. Like, if there's going to be an intern, it's going to be someone local, inevitably. Like, so that's just kind of the way it is. Like...
What? I got people flying in from Austria.
Yeah.
Well, yeah, but you also own a fairly large house with space to host them. And
that's true.
And are a very particular peanut in the sense that, like, you'll just have
I can put up with people's bullshit.
Yeah, at a level that would yeet them out a window, like.
Yeah.
So that's the thing, but yeah, I mean, but the other thing is, like, you got to keep in mind there is a pandemic going on right now, so just showing up at Gabe's house is probably not a good idea.
That's okay. I've got a UVC light. I'll just irradiate you.
I'd be sterilized off the plane.
Yeah, unless you want to be tested on. Don't just show up at Gabe's house. Yeah, so that's kind of it. I think we're gonna wrap it up for the day.
Yeah.
So yeah.
Well, as always, it was a great time.
Yeah, this was a lot of fun. We'll probably do another one of these next week. There'll be another Who's Gene on probably Tuesday. I think I'm gonna end up having there'll be a poll that goes up probably Sunday or Monday, depending on when I settle on topics I want to choose from. That'll go up on the channel page here on YouTube. It'll probably also go up on Twitter. We'll pick a topic from there, and then that'll set the topic for Tuesday. If you have topics that you want me and Gabe to talk about next week, be sure to leave them either in the comments here, not in the live chat, like in the actual comments under the video once this is up, or just send them to me on Twitter. Both work. We've kind of just been, like, picking whatever seems like a good thing to chat about, but, like, if there's a specific thing
What are you excited about today?
Bees!
All right,
yeah.
then. Let's
Bees
talk
it
about
is. So, if there's a thing that you specifically want us to talk about, leave it to us on Twitter. Other than that, thanks for hanging out. This has been a lot of fun. Thanks to the couple people who donated. It really is greatly, greatly appreciated. And other than that, I think we'll wrap it up for the day. So,
Yep.
Again,
Thanks to everyone.
Thanks everyone for hanging out. It's been a lot of fun, and I will see you on the next one. Bye-bye.